Hi guys, we're back. I'm Jade, I'm a sophomore, AP Biology. I think you get the gist if you watched my first video. I took a shower, I finished my energy drink, I'm working on a Mountain Dew right now, and that's hopefully gonna keep me going. I noticed that my last video is kind of long in regards to how many chapters we do have to cover, so I'm gonna try and keep this to 10 minutes or under, allow you guys to really just get the gist of everything, but not in as much detail, I guess. I'm going to try and talk fast. I'm going to try and go through these concepts without forgetting things. And yeah, that's it. We should get right into it. Chapter 3 in the Cliff's Notes book is about cells. It's the structure and function of the cell. It's talking about the phospholipid membrane, which we covered a little bit in last video, and how it's selectively permeable. This means that certain things can go in and out, and certain things can't as easy without channels or protein carriers, different things like that. So this is talking about how the lipids are hydrophobic, so water can't go through as easy, but different gases, uncharged molecules, yeah, they can go through pretty easy. Then we have proteins that are throughout the plasma membrane, and they provide a different range of functions. We have channel proteins, ion channels, carrier proteins, transport proteins, recognition proteins, receptor proteins, and adhesion proteins. I haven't seen a lot of those in my reviews, but I do think that channel proteins are important. They provide open passageways through the membrane for certain hydrophilic molecules. So those will be water-loving molecules. They're polar. And then we also have carrier proteins, which bind to specific molecules and then are transferred across the membrane after a change of shape that allows it to sort of adhere to the molecule that they're transporting across, and that helps it get in or out of the cell. Then we have receptor proteins, and they're binding sites for hormones or trigger molecules, and then those provide signal pathway that will trigger a response within the cell most of the time. The other ones I haven't really seen a whole lot, so I'm gonna not go into detail on those. You can always read about them in your Cliff's Notes book or look up proteins in plasma membrane and find a video on that. Now we're going to go into organelles. There are many of them, and they have certain functions. I'm going to give the gist of them and give you the main points that you should remember. The nucleus contains all of the genetic material. We're talking DNA, your chromosomes, all of the things like that. That is its main purpose. You also have the ribosomes, and basically, when they're in the cytoplasm, they assist in the assembly of amino acids into proteins that we covered in the last unit, and that's a different process that we're going to cover in a later video. Then we also have the endoplasmic reticulum, which is also called the ER. We have the rough ER and the smooth ER. The rough ER is going to create glycoproteins by attaching polysaccharide groups to polypeptides as they're assembled into ribosome, or by the ribosomes, sorry. So you can think of this as sort of production assembly of proteins. And then you have the smooth ER, and that does not contain ribosomes. The rough ER has ribosomes attached to it. The smooth ER is going to be the synthesis of lipids and steroid hormones, and it's also going to be the breakdown of toxins and drugs. So, excuse me, you're going to see a lot of smooth ER in areas like the liver. The Golgi apparatus, that's going to collect and modify the proteins and lipids made in other areas of the cell, such as the smooth and rough ER, and it's going to package them into vesicles that are either going to be transported out of the cell or used within the cell. Got it, okay. Lysosomes are going to contain hydrolytic enzymes within them, and those enzymes are going to be used to break down food, cellular debris, foreign invaders. They're going to recycle cell material. That's their main purpose. Then we have mitochondria, and these are so important. They aid in aerobic respiration, so respiration with oxygen that we'll cover in another video, and yeah, that's basically its main process. It's the energy powerhouse of the cell. It provides the cell with energy, and it is a catabolic reaction, I believe. Yeah, I think so. Chloroplasts are going to carry out photosynthesis in plants, not animals. There's other things that do photosynthesis other than plants, but when you think of photosynthesis, you don't want to think of humans. We don't do that. Microtubules, intermediate filaments, and microfilaments help in establishing the shape or movements of the cytoskeleton, which is kind of just like a cellular skeleton. You think of our skeleton as our bones holding us together, keeping us in shape, and that's exactly what the cytoskeleton does for cells. Then we have the vacuoles and vesicles, which are fluid-filled membrane-bound bodies. So there's many different kinds. I'm going to talk about transport vesicles, which are going to move materials between organelles, and then there's secretory, I don't know how to say that, vesicles that move materials from the Golgi apparatus to the plasma membrane and eventually out of the cell. I think those are two important ones that you should know. And then you have the cell walls that are found in plants and fungi, and many protists, and that just helps keep the structure of the cell. Animals do not have cell walls. We don't. We have membranes, so do plant cells, but we don't have cell walls. And I think that's about it for organelles. Those are the main things that you should know. Now, we need to talk about prokaryotes and eukaryotes. I apologize for all of my stuttering. I'm just trying to get through this as fast as I can and give you as much information as I can. It's difficult. So, prokaryotes and eukaryotes. You need to know that eukaryotes developed from prokaryotes. Yes, I believe that's correct. But the differences are that prokaryotes do not have a nucleus. Their genetic material is kind of free roaming. Also, the hereditary material in prokaryotes are single, naked DNA molecules without the proteins that are associated with DNA in eukaryotic chromosomes. We also want to note that prokaryotic ribosomes are smaller than those of eukaryotes. That's about it. They both have ribosomes. Prokaryotes just have smaller ones. And then the cell walls of bacteria are constructed from different things than the cell walls of archaea. And that's about it. That's about all it says. And we're going to skip the last one because I don't think it's important. It talks about flagella, and we didn't cover that in the organelles, so we're just going to skip that part. But you can always look up other videos, like I said. Now we're going to talk about movement of substances. So bulk flow, I want to talk about that because I found that and I thought it kind of makes sense if you just think about it logically, but I couldn't pinpoint what it was. So it's basically the collective movement of a substance in the same direction in response to a force or a pressure. So a good example of this would be blood moving through a blood vessel. That's bulk flow. Then we're going to talk about hypertonic, hypotonic, and isotonic. So isotonic, it's the same. It's like an isosceles triangle. You have two sides that are the same. But in this case, it's just an equal concentration of solutes relative to another region. And then a solute can also be hypertonic, which means basically a higher concentration of solutes, so hyper, higher. And then hypotonic is a lower concentration of solutes, so hypo, lower. And this is in reference to solutes, not water, not any other thing. We're talking about solutes here, not solvents. Passive transport. Passive transport, you can think of simple diffusion. It doesn't require energy, and it's just movement of things from a higher concentration to a lower concentration. And that's how you want to think of diffusion most of the time. Things do move against concentration gradients sometimes, but most of the time that requires an expenditure of energy, which doesn't happen in simple diffusion. Osmosis is basically just diffusion of water. You want to keep that in mind. And then plasmolysis is the movement of water out of a cell that results in the collapse of a cell. So that's going to be different. This is the collapse of a cell due to the movement of water out of a cell. And that's also cell lysis, or can reference that. It occurs when swelling of the cell causes the cell to burst. So then we have facilitated diffusion, and it's basically diffusion that happens through channel proteins, and it doesn't require an expenditure of energy. And then we also have active transport, which is the movement of solutes against a gradient. So you're going from lower concentration to higher concentration, and because of this, you're going to need an expenditure of energy. Basically, think of this as like, I'm a runner. So when I go running, if I'm going with the wind, right? If the wind is pushing me from the back, I'm going this way, then that's just going to be simple diffusion. It doesn't require very much energy on my part. I'm just going with it. But then if I'm going against the wind, it's going to be a lot tougher for me, and it's going to require more energy. So that's how I think of active transport. And then there's also different things that I want to go over in terms of vesicular transport. Uses vesicles. I don't know if I said that right. Exocytosis is things moving in vesicles out of the cell. Exo, out. And then you have endocytosis. So that's bringing larger molecules of things into the cell. Endo, enter. So phagocytosis is a form of endocytosis in which undissolved materials enter the cell. And then you also have pinocytosis, and that is like phagocytosis, but it's a form of endocytosis in which molecules enter the cell, and they are dissolved. And then we have receptor-mediated endocytosis, which is just another form of pinocytosis, and it's when specific molecules in the fluid surrounding the cell bind to specialized receptors that allow them to come in. And that's all I have for you for this video. I hope that was helpful.